Good evening. I'm Darla. I'm the director of the supportive care program. You probably have seen my picture in our magazine. Anyway, tonight you're here to see Dr. Sledge talk about the new biology of breast cancer and its therapeutic implications. Many of you know Dr. Sledge. He's the chief department. I knew I was going to blow this one. He's the chair of the Department of Oncology here at Stanford. So we're quite honored to have him come and talk to you. My colleague Esther, many of you met on the way in, will introduce everybody again at the end. You can ask questions throughout the presentation if you'd like to. If you have a burning question you want to ask Dr. Sledge, please don't hesitate to raise your hand and he'll answer your questions. Speak as loud as you can. We are videotaping this so that in a few weeks it'll be on our website, all right, so that you can look at it again. So without further ado, I'd love to invite Dr. George Sledge to come on up here.
So thank you, and thanks in particular for coming tonight. I always cringe a little bit when I hear that I'm being photographed, because I consider myself the least photogenic person on the planet. I was actually telling one of my faculty earlier today, I have exactly one picture that I like. And that's because when I was chief of the American Society of Clinical Oncology about 6 years ago, a photographer followed me around for an entire week, took hundreds of pictures, and I finally found one that I liked. So, so I would not encourage anyone to go on the website and look at me. Anyways, it's a delight to be here. What I'd like to do tonight is pretty simple. I'd like to, first off, convince you that breast cancer isn't a single disease, but rather that it's a family of diseases. And then I'd like to introduce you to some of the members of the family. And this, by the way, is a really dysfunctional family, okay? And we all have relatives like this, right? But as we've learned more and more about the family members, we've learned more and more about how to treat them. And we've gotten, I think, better and better over the years, in large part because we've learned about cancer biology. And I suspect that we're going to get even better going forward. Now, the talk is set up in such a way that you will not offend me if you interrupt. So feel free to ask questions. If I say something that doesn't make sense to you or something you find obnoxious, feel free to interrupt and exclaim, and I will try and repeat myself and be a little bit more intelligible. So let's start here. This is a slide that if you're a breast cancer doctor, you've seen about 5,000 times in the last decade. And it actually points to some research that was done here at Stanford in the late 1990s by a group of molecular biologists. Now, in the past, in breast cancer, if you go back 20, 30, 40 years, we knew a little bit about breast cancer biology, but not a whole lot. And the great excitement that breast cancer researchers and breast cancer doctors have is in large part due to the fact that we've learned an awful lot about the biology. And some of what we learned actually was learned here at Stanford about 20 years ago. The investigators took a bunch of breast cancers and ground them up, and then did the very first molecular portrait of breast cancer. And what they did is you took the DNA from the breast cancer and you applied it to a certain test. It was called an Affymetrix chip. That doesn't matter. And then the information that came out of that was fed into a computer. And of course, the computer knows nothing whatsoever about breast cancer. But what the computer is very good at doing is clumping data. And so when it clumped data, what it showed was that basically breast cancer represented several fairly distinct entities from a molecular biology standpoint. The DNA was just different. And they were given names like luminal A, luminal B, basal-like, ERB2-like, and normal breast-like. The names aren't particularly important. What's important is that this was the very first evidence that we ever had, for strong evidence at a very basic genetic level, that breast cancer was not a single disease, but rather a family of diseases. Okay, and this turns out to be really, really important. Now, if we look at what we know in the clinic, these link up pretty well with what we know from a molecular biology standpoint. So if we look at all of breast cancer and break down into subtypes, there's a fairly large group of breast cancers, and this is about probably 2/3, 3/4 of breast cancers, that are characterized by being sensitive to estrogen, to female sex hormone. And these tumors in general are positive for estrogen receptor. There's another group of tumors, and this represents about maybe 15 to 20% of tumors, that are positive for a protein called HER2, more about that later. And then there's a third group of tumors which has a bunch of different names. Basal, basaloid, the other term you'll hear is triple negative, which represents another fairly distinct group of tumors. And it turns out that these are not just molecular biology constructs. These are things that are very important in the clinic, because just as in your families, family members can act very, very differently, okay? Some of them are kind of slow and not particularly aggressive. Some of them are total twits and you don't want to be in the same room with them, okay? And the molecular biology has given us a pretty good idea of who these are. So let's look at some of the family members, okay? So the first one is the one that we would call estrogen receptor positive. Now, in molecular biology terms, these are called luminal breast cancers, but let's just focus on the estrogen receptor positive, okay? And the way this works has been known actually for a pretty long time, actually dating back to the late 1960s, early 1970s. What was discovered was that if you looked at female sex hormone, estrogen, estrogen freely crosses into normal cells and into breast cancer cells. And then once it's inside the cell, it hooks up with what's known as the estrogen receptor. And once it hooks up with the estrogen receptor, that then goes to the cell nucleus, the brains of the cell. And that in turn turns on a whole bunch of different things. And estrogen turns out to be, as men have known for a long time, incredibly potent, okay? Because it regulates something like 700 different genes in your body. I mean, you have about 21,000 genes that make up all of the genes in your body. 700 of them are regulated by a single protein, by estrogen. Kind of cool, okay? Among the things that happens, though, is that when you toss estrogen in with an estrogen receptor, it tells cells to grow and divide. And this is particularly important if we're talking about a malignant cell, because the cell will grow and divide and then now spread, okay? Now, the good news is that you can block estrogen's ability to do this in a number of different ways. And of course, I'm sure many in the room are familiar with some of these ways. We have pills that block estrogen directly, like Tamoxifen. We have pills that lower estrogen levels in the body, like aromatase inhibitors. We have shots, like Faslodex, that tell the estrogen receptor to basically stop working. We have a bunch of different ways, and then surgically, of course, we can remove the ovaries to lower estrogen levels. What all of these things have in common is that they're basically telling the estrogen to stop signaling the estrogen receptor and telling it to stop cell division, cell growth, okay? Now, in the clinic, we've known this for a very long time, and we've done bunches of studies over the years. And in those studies, what we have discovered is that if you look at using a very old drug called Tamoxifen, that if you give Tamoxifen to patients with early stage breast cancer, we significantly reduce recurrence of breast cancer, and we significantly reduce the death rate due to breast cancer. Now, Tamoxifen is a very old drug. We actually have better drugs available now. And we use Tamoxifen for longer than we used it in the studies that make up these charts. But again, what we've known is that for a very long time, certainly over 20 years now, that we can lower breast cancer death rates in patients with early stage breast cancer by blocking estrogen. Now, if you looked at those curves closely, you notice that we didn't cure everyone, okay? And we didn't cure everyone because cancer cells are smart, and cancer cells are pretty good at finding ways of getting around the roadblocks that we throw up. And so doctors have, for many years, tried to get some idea of why this is the case. And this is a very confusing figure, so don't look at it, just listen to me, okay? it turns out that when we throw up a roadblock, think of this, think of the cancer cell as a criminal that's trying to escape town, okay? And imagine that you throw a roadblock up on the 101, okay? Well, the cancer cell can still get out on the 280, right? Okay? And so if we look at what happens in breast cancer cells, that phenomenon also is the same. We can throw up one roadblock, but the cancer can find another way around it. Frequently, the way it finds the way around it is to use a different growth factor that also signals the breast cancer cell to divide and grow. And for molecular biologists, the term that they use is crosstalk, which is to say the receptors are talking to each other. And so if you block one, the crosstalk can still make the cancer cell divide and grow. Okay. By the way, medicine is full of jargon, but actually the ideas are pretty simple if you think about it. Okay. Now, as it turns out, since we've been able to identify the sources of this crosstalk, we can actually develop drugs that interfere with crosstalk. And there's a drug on the market for patients with advanced breast cancer. It's called Afinitor or everolimus, which inhibits crosstalk. And so we've been able to use this knowledge to actually improve treatment for patients with advanced disease. And if you look at this in clinical trials with this drug that inhibits crosstalk, what we see is that it significantly lengthens the period of time that patients remain progression-free for their disease. Now, one of the other things that we've looked at in terms of the estrogen receptor goes to the question of cell division. And I mentioned that one of the things that estrogen does is that it tells cancer cells to divide. And for a long time, what we knew about breast cancer biology was estrogen hooked up with the estrogen receptor and then something happened, okay? Not very sophisticated, right? Okay, but what we found subsequently is that there's a whole pathway after estrogen hooks up with the estrogen receptor, and that by pushing things along this pathway, you get the cancer cell to start dividing. So I'll leave this up for another 15 seconds so you can memorize everything here, okay? Or if you prefer, I can give you a homely analogy, okay? Which is the way I think of it, okay? So think of estrogen as coal, and think of the estrogen receptor as the furnace that you're throwing the coal into. And think of this, so we're on a freight train now, okay, a coal-fired freight train, okay? So we're throwing the estrogen into the furnace, okay? Is that what moves the train down the track? No. You have gear, you've got all sorts of gears and pistons that are actually attached to the wheels and they're moving them down the track, okay? So if we keep the coal from going into the furnace, that's good. But if we throw a spanner in the works, that's good too. So the combination of those two things, removing the coal, throwing the spanner in the works better than just removing the coal. And that's what we found out both in the laboratory and in the clinic. And that's led to the development of a number of new drugs with really awful medical names like palbociclib, abemaciclib, and ribociclib, okay? They all end with cyclib, and it's cyclib because they're preventing the cycling of cancer cells, okay? And all of these three drugs have come on the market within the last 2.5 years. And what collectively they have done for patients with advanced breast cancer is to roughly double the length of time that before a patient's tumor starts growing again. It's a major advance, not a cure for patients with advanced disease by and large, but one that can give many, many months, in some cases, many years of useful additional quality life. We're now taking these drugs and moving them into patients with early stage breast cancer in the hopes that it will be curative in that setting. And at Stanford, we've been participating in a couple of large clinical trials that are national trials, looking at the first of these drugs to come out to see whether or not we might, in fact, be able to do a good job in terms of preventing recurrence of the breast cancer. So an exciting prospect. Yes, please. I saw a hand back there.
If I may ask, why is it limited to HER2 negative only?
Great question. One of the things that was done when these drugs were, so the question is, why HER2 negative estrogen receptor positive tumors? It's a great question. So when these drugs first came out, they were first looked at in the laboratory, and what they did in the laboratory was to take a whole bunch of different breast cancer cell lines, where people had taken bits of tumors and then grown them in test tubes. Some of these cell lines were estrogen sensitive, some of them are HER2 positive, and some of them are triple negative. And what they found when they did that was that the cell lines that were the most sensitive in terms of the spanner and the works drugs were the ones that were estrogen sensitive breast cancers. Okay? Now, the triple negative ones by and large didn't benefit at all. Some of the HER2 positive ones did, particularly if you had a tumor that was estrogen receptor positive and HER2 positive. And that's led to clinical trials that are going on right now for patients with advanced breast cancer to see if it'll work there. So ask me that question again in 2 years and I might have a different answer for you. Okay? Because my suspicion, and the suspicion of a lot of people who've worked with these drugs, is that these drugs may work well for some HER2 positive tumors as well. Okay? Great question. Okay. Anywho, as I mentioned, we've looked at these drugs in terms of many clinical trials. I'm partial to this particular trial. It's called Monarch II because yours truly was the principal investigator for the trial on a global basis, and it got the drug approved by the FDA 2 weeks ago. So I'm not a little bit biased, okay? But basically, this was a trial that took patients and randomly assigned patients either to get the CDK4/6 drug, abemaciclib, or to get a sugar pill, a placebo, and then looked in terms of how patients did. And basically what we saw was that patients who got the new drug did a whole lot better than the patients who got the old drug. And so this has led to the approval of this drug literally a couple of weeks ago by the FDA. So for estrogen-sensitive tumors, we're making some progress. We're not all the way there yet. I think all of us believe that we've learned a whole lot more about the biology, and I think all of us in the field believe that we're going to, a few years from now, we're probably going to be doing measurably better in terms of preventing recurrence. That's our strong hope. I'm a great believer that research cures cancer. I will flat out tell you that the most courageous people on the planet are not policemen or firemen. They are women who go on to breast cancer clinical trials. Okay, and those women are my heroes. So let's move on to HER2. Okay, so HER is not has no gender identity here. Okay, HER is short for human epidermal growth factor receptor. Okay, and this is a family of receptors, and these are basically proteins that are found on the surface of the cancer cell. There's 4 members of this family. HER2, which is this kind of pretty one here, is the one that's most common in human breast cancer. It's present in about 15–20% of all breast cancers. And we know that if a tumor is positive for HER2, that will drive the growth of the breast cancer and will increase the likelihood that the cancer will spread outside the breast to lymph nodes and to distant sites of disease. And we learned this in the late 1980s. And by the time the mid-1990s had rolled around, we started seeing the first drugs to treat HER2-positive breast cancer. And certainly the best known of these even today is the one that came out in the 90s, one called Herceptin. And Herceptin is an antibody. Antibodies are normal proteins that your body makes to ward off viruses, bacteria. But this is an antibody that's made in the laboratory. It's actually made in what looks like a fermentation vat. I'm from Wisconsin, so it reminds me ever so much of breweries that I saw growing up in Wisconsin where they make these antibodies. They take the antibodies, isolate them, concentrate them, we inject them into the body, and then the antibody circulates in the blood, finds the breast cancer that's positive for HER2, and attacks that cancer. Okay, and that's what antibodies do. And so we've had since the 1990s antibody therapies for metastatic breast cancer, then subsequently for early stage breast cancer. Now again, I like to keep things simple. This is how I remember things. Okay, and if you think of this as a catcher's mitt, show of hands for baseball fans. Okay, good, great, all right. I made the mistake of showing this slide in Europe, and no one had a clue what I was talking about. Okay, okay. But if you think of this as the receptor is a catcher's mitt, and the catcher's mitt is capturing what's called a ligand. Okay, the ligand is the signal that once it's been captured in the catcher's mitt, tells the cancer cell to start dividing and growing. Okay. And so HER2 is a receptor on the cell surface. It's a catcher's mitt that's capturing the ligands. Now, what turned out to be really, really important is that we could measure this. And I say it's really, really important because if you think about all drugs have side effects, and not all drugs work for everyone. But if you know what the drug is supposed to be working on and in whom, you're much more likely to get benefit, and you're going to get a lot less side effects. The best way to avoid side effects is to not give a drug to someone who won't benefit from it, right? Okay? So in the clinic, what we can do is we can take a part of a patient's tumor, and this can be either the primary tumor in the breast, or if the patient suffers a recurrence elsewhere in the body, a piece of a tumor from the liver or the lungs or the bones, and we can stain it. And by staining it, we can tell whether or not the tumor is going to be sensitive or not sensitive to HER2-targeted therapy. And by the way, to anyone who ever thought that science was really, really sophisticated, high-tech, really cool, gee whiz sort of stuff, here's what the pathologist does, okay? You see the little blue dots, and you see the little pink dots. So what you do is divide blue by pink, or rather pink by blue in this case, okay? And in a normal cell, you have 2 copies of the pink dots and 2 copies of the blue dots, so the ratio of them is 1, all right? But in a HER2 amplified cell, what we see down here on the right, if the ratio is more than 2, we call that a HER2 positive cell, okay? Very high tech, right? Okay, second grade division, okay, high tech level, okay? And this is what's called a FISH test. We also can use something called immunohistochemistry, but this is what the doctor uses basically to say, should I be treating a patient with something that targets HER2? And we've known since about a decade ago that if you look for patients with early stage breast cancer, and by early stage I mean patients who don't have obvious metastatic disease, and you treat them with an antibody to HER2, you can roughly cut in half the risk of recurrence and death due to HER2 positive breast cancer. Now, we keep learning new things, and actually one of the, for me, I'm someone who's, like most scientists, I'm driven to a huge extent by idle curiosity. If you put me in a boring meeting, after about 2 minutes, I will zone out and I will start writing down ideas on a piece of paper. I believe strongly that in the long run, cancer is going to be cured by a couple people who run into each other at a water fountain. Okay, all right, because that's where the real science is done, is a couple people bouncing ideas off of each other. And so a lot of what we've learned in recent years is a result of scientists being curious about how something works. So I gave you the story of HER2, and it's a nice story of there's this receptor, and it's like a catcher's mitt, and if it catches something, it tells the cancer cell to divide and grow. But of course, it's more complicated than that. And I showed you a few slides ago that HER2 was part of a family, okay? And they're called HER1, HER2, HER3, HER4. It turns out that 2 of these receptors have to come together to signal the cell. So if you think of the cell surface, like this one here, as having these receptors sort of spread out on the surface, what happens is that if two of them come together, if they hook up, the scientific term is if they dimerize, but think of it as two of these guys hooking up, that's what signals the cell to grow. And it turns out that the favorite hookup for HER2 is with its relative HER3. Okay? All right? So, okay. Think Bonnie and Clyde. Okay, show of hands for those who have seen Bonnie and Clyde. Okay, good. All right, the rest of you need to get a life. Okay, all right. So, a true dysfunctional relationship. Okay, when these two got together, they caused a lot of trouble. And that's what happens in a cancer cell when HER2 and HER3 hook up. Okay? But we can develop drugs that prevent hookups. And so one of the drugs that's been developed in recent years is a drug called pertuzumab. You may have heard of it as Perjeta. Perjeta works by preventing hookups. It's you know, the chaperone that comes between the two and says, Bonnie, Clyde, stay away from each other. It'll only be misery if the two of you get together. All right? And so when we do that, and add that to Herceptin, and pertuzumab, like Herceptin, is an antibody, we can further decrease the growth of the cancer cells and prevent a lot of HER2-positive breast cancer cells from growing and spreading. And the way that we learn these things in the clinic is to do large clinical trials. The trial that proved this was a trial with the lovely name of Cleopatra. Oncologists, by the way, love trials with pretty names, okay? And so we have Cleopatra here. One of the early HER2-positive trials was called Hera. We sooner or later we're going to run out of Greek goddesses and ancient figures to name it after, but pretty name anyway. This trial took women with advanced breast cancer, where under normal circumstances they would get the combination of chemotherapy plus Herceptin. Please, come on in. Nothing to be sorry about. Please, come on in. Have a seat. And compared it to the same plus Perjeta. And this was a trial that was done around the globe. And what we found was that the simple act of adding this other antibody added close to 1.5 years of life for patients with advanced HER2-positive breast cancer. When I look at these survival curves, I am alternately depressed and delighted because I've been around for a long time. If I were to show you a survival curve from 1998 for HER2-positive breast cancer cells in the metastatic, HER2-positive disease in the metastatic setting, in 1998, before Herceptin came around, I told patients with HER2-positive metastatic disease that on average they had about 16 months to live. Okay, so over the last 15, 20 years, we've gone from 16 months to 56 months. We've added a lot. And indeed, we have some patients with advanced HER2-positive disease who are alive and free of disease 10, 15, 20 years out at this point. But we still obviously have some work to do. We have a number of other drugs that target HER2 that are coming along fast, and which hopefully we'll add. One of these is a drug called TDM1, and I mention this because it's the first of a new class of drugs, and I think it's kind of a clever thought. So, as I mentioned before, if we look at trastuzumab, Herceptin, this is a drug that it's an antibody, it floats around, it binds to the cancer cell. And we thought of it first of, let's use the antibody to shut off HER2, and that'll put the cancer cell to sleep, it'll stop growing. But there's another way that you can use it, and that is to use the antibody as a delivery truck. And you can use it to deliver poison just to the cancer cell. And this is sort of, if you think of it, sort of a smart bomb, just attacking the cancer cell. And what was done, and this was done just a few years ago, was to take the antibody and to bind it to a plant poison, This is the DM1 that you see here in TDM1. The T is the trastuzumab or the Herceptin. DM1 is a very old plant poison. It was actually looked at as a cancer drug back when I was an oncology fellow 2 years ago. Not quite, I wish. Back in the late 1970s and early 1980s. And it was a great drug for killing cancer cells. I mean, it was a world beater for killing cancer cells in the test tube. The problem was when you put it into a patient, it rotted the liver, okay? And so it went on the shelf for close to 3 decades where no one looked at it until someone finally said, what's the most toxic thing we can find that we couldn't give in the past but is really great at killing cancer cells? And let's add it to trastuzumab, let it take it just to the cancer cell rather than to the liver and see what happens. And what happened was, when we ran the clinical trials, it was less toxic than giving a HER2 targeting agent plus a chemotherapy drug if you gave them as separate drugs, okay? And here's the best part, it wasn't just less toxic, it was more active. People live longer. That got the drug approved by the FDA. So pretty cool, yes.
So it is approved in use?
Yes.
And if you've already had a first line of treatment in which you've been on Herceptin and Perjeta for a while and they maybe have stopped working,
Yeah.
this wouldn't work anymore then?
You could then use this drug, TDM1, yes. Yeah, and indeed, that's exactly where we use it, okay? Okay, so let's move on to the third dysfunctional member of the breast cancer family, and this is so-called triple-negative breast cancers. And we call them triple-negative because they're negative for estrogen receptor, they're negative for another receptor called progesterone receptor, and they're negative for HER2. Now, if you think about it for even a second, this is the dumbest description you've ever heard of in your life, okay? Because it doesn't actually tell you anything about the cancer, right? Imagine you're walking down the street and you see a red-haired woman, and you're asked to describe that woman, and your description is non-blonde, non-brunette, and doesn't have a beard, okay? All right? That's triple negative, okay? All right? So one of the most powerful things that we do in science is we name things. And the very naming of things is the first step towards understanding them. And so triple negative was a dumb name. And it's taken us a decade or so to start thinking of it not in negative terms, but in positive terms. But if you look at these tumors, the pathologist would say that they have high nuclear grade and they're highly proliferative. In other words, they look ugly under the microscope and they grow fast. Interestingly, this is the family home for patients whose tumors have a BRCA1 mutation, or actually who every cell in their body has a BRCA1 mutation. Most BRCA1 mutant cancers are triple-negative breast cancers. We'll get back to that in a minute because it turns out to be really important in terms of some of the new treatments that are coming along. These cancers tend to be fairly sensitive to chemotherapy, but at the same time, the cancers are really, as I said, they're really ugly cancers, so they're pretty good at developing resistance to most of the chemotherapy drugs eventually. And it turns out that some chemo drugs work better in triple negative than others. And so I'll tell you a bit more about that as well. But the main thing about these triple negative breast cancers is that from a genomic standpoint, if you look at the DNA of these cancers, these are pretty chaotic cancers. They pick up lots of mutations. Sometimes they pick up lots of mutations because they have a BRCA1 mutation, and BRCA1 mutants pick up a lot of mutations. But for whatever reason, as a family, most of these are fairly highly mutated. And highly mutated equals smart if you're a cancer cell. So that's what makes these dangerous. Now, we just started to look at these cancers over the last decade or so. It's interesting, if you go into the medical literature and type in the National Library of Medicine's PubMed service, if you type in triple-negative breast cancer, the first time you ever see the term in the medical literature is 2004. Okay. So we didn't even think about that. We didn't even give them a name till 2004. Even if it was the wrong name, we didn't even have a name for this until 2004. And if you haven't named something, it's unlikely that you're going to be studying it, or studying it as a specific entity. So this is something where it's just in the last decade or so where we've actually started studying these in a major way. Some of it has been studying things with some old drugs. And so 2 old chemotherapy drugs, carboplatin and docetaxel. Our drug, docetaxel is Taxotere. These are drugs that we use all the time for breast cancer. But when we give someone chemotherapy, one of the shameful things, I say as a medical oncologist, one of the shameful things about medical oncology is that our approach to giving chemotherapy is to throw the spaghetti against the wall and see what sticks. Okay, we're not very good at predicting which drugs work for which patients. So one of the ways of looking at that is actually to do a study, and this was done in patients with triple-negative breast cancers. It was given the lovely name of the TNT trial, the triple-negative trial. Okay. And patients were randomly assigned to 1 of 2 chemotherapy drugs, because chemo has been what we've had available for patients with triple-negative disease. One of these drugs is a drug that damages DNA, and the other is a chemotherapy drug that works by a different mechanism. It's not damaging DNA. So what happens when you throw these drugs in? Well, it turns out that if you have a BRCA mutation, either a BRCA1 or a BRCA2 mutation, carboplatin works a whole lot better. Now, carboplatin, again, is the drug that damages DNA. So if you have a triple-negative breast cancer and that triple-negative breast cancer has a BRCA mutation, you're much more likely to respond than if you don't have the mutation. Now, for the rest of the triple-negative for the rest of the patients, it doesn't make a whole lot of difference. Both drugs work equally well, but if you've got this particular mutation, you're more likely to benefit from a drug that damages DNA. Now, this has led to other ideas, and this has led to something called PARP inhibition, okay? And a whole new class of drugs. Now, if you look at patients who have a BRCA mutation, one of the questions that scientists have asked for a long time is, why does having a BRCA1 or BRCA2 mutation give you cancer? And it turns out that it gives you cancer because these are normal proteins in the body that work to fix DNA when it's damaged. And so if you have a BRCA mutation, what happens is that you're not very good at fixing the DNA inside of normal cells. And the end result of that is that you get a whole bunch of mutations and cells start to become cancerous. Okay. Now, it turns out that BRCA1 and BRCA2 are part of the fix-it kit for the cancer, but it turns out there's a whole lot of other things that help fix DNA when it's damaged. Last time I saw, there was something like 17 different proteins that fix DNA when it's damaged. I'm sure the number's up over 20 now, but I lost track a few years ago. So what if we, in a patient who already has inability to fix mutations due to one thing, what if we shut off something else, another part of the fix-it kit? And the one that's been looked at is something called PARP. I could share with you what that acronym stands for, but I don't want you all to fall asleep in the middle of my talk. If we shut off now 2 things in the fix-it kit, it turns out that makes the cancer cells much more sensitive to drugs that damage DNA, like carboplatin. Okay? So this, again, is how learning biology allows us to do better things. So think of this. Think of the DNA damage repair toolkit. Every cell in your body has tools and a bunch of different types of tools to fix DNA. By the way, you can buy this at Target, okay, in the lovely pink color. I found this online. And if you think about, show of hands for the do-it-yourselfers in the audience, people who actually fix things around their house. Okay, good, great. Okay, I pretend to. And then I ask my wife, You probably should call a repairman on this. But if you think about, let's say something's broken in your house and you want to fix it, and you've got a repair kit. And I don't know about you guys, but I'm always losing something for my repair kits. Okay, some screwdriver or wrench or something goes walking for some reason after I've used the kit. But frequently there's still something else in the kit that you can use to fix it. But if you start taking several things out of the kit, all of a sudden you can no longer fix it. Okay, and this is the idea behind the new drugs. Let's take some more things out of the fix-it kit and make it impossible for the cancer cell to fix itself. Okay? And this has led to a trial called the Olympiad Trial, just came out 3 months ago. Very first trial in patients with fairly far advanced triple-negative breast cancer. But what this trial did was basically to compare a whole bunch of old chemotherapy drugs that we've used for years with this drug that takes another tool out of the DNA repair kit. And it turns out that when you do the comparison, the new drug, the drug that takes the repair kit, that messes with the repair kit, allows patients to go longer without the cancer progressing. And it also turns out that it's less toxic than our standard old chemotherapy drugs. Now, myself and many of my colleagues, when we looked at this data, we weren't hugely impressed, I must say, because this is in patients with fairly far advanced breast cancer. It added a few months, but I know the patients in my clinic who have advanced breast cancer want more than a few months. Okay, I mean, they want years or decades. They have kids at home that they want to see graduate from college and get married. Okay, so can we do better than this? And the answer is we're looking to see whether or not there's other toolkit things that we can work with. And can we now actually take the drugs, what are called PARP inhibitors, and actually combine them with other drugs and actually do a whole lot better? Now, there's some reason to believe that this approach may work pretty well in some settings. And the best evidence for this comes not from breast cancer, but from ovarian cancer. And ovarian cancer, like breast cancer, is a cancer where we see a lot of these BRCA mutations that cause both diseases. And in ovarian cancer, if you look at a patient who has ovarian cancer, the standard way of treating ovarian cancer, and most ovarian cancers present as stage 3 or 4 disease, the standard way of treating them has always been to give patients a few treatments of chemotherapy with a platinum-based drug, the DNA-damaging drug, and then to stop and then wait until the cancer comes back, and then to start treating again, which has always struck me as kind of frightening if you're a patient, the sort of watching and waiting thing. So in ovarian cancer, what was done was a trial in which patients got the standard chemotherapy treatment for ovarian cancer, a few cycles of chemotherapy with a platinum-based chemotherapy regimen. And then the patients were randomly assigned either to get the PARP inhibitor or to get a sugar pill, a placebo. And that trial showed that on average, a patient who just got the chemotherapy on average went about 5 or 6 months before the tumor started growing again. If you add in the PARP inhibitor, it now goes up to about 26 months. Okay? And so that's pretty exciting. So are we going to be able to do something like that in breast cancer in the near future? There's a whole bunch of studies coming down the pike. This one I'm showing you here is the very first one. This is hot off the presses. This just came out 3 months ago. But there's a whole bunch of other trials coming behind it. And what we're hoping is that we're going to be able to do ever better as we learn more about how to use these agents. Please, yes?
How are you keeping the PARP inhibitors from breaking other cells, healthy cells?
Great question. So the question is, all right, you're tossing in these things that mess with the DNA damage repair. How come it's not killing you? I guess is the question, right? And the answer is that cancer cells are different than normal cells. So part of how you get a malignancy is that someone who has a BRCA1 mutation, they're born with it, it's defective, but you have 2 copies of BRCA in every cell. You get one from your mom and one from your dad, okay? What happens in the cancer cell is that you end up with more than one mutation there. So most of your cells in your body actually have one normal copy and one abnormal copy. It's the cancer cells that get into trouble, okay? All right, let's talk about immunotherapy, because this is something I'm asked about all the time in my clinic. And the answer in breast cancer for immunotherapy is that we're not as far along yet as we are for many other cancers. Now, in recent years, there's been an explosion of new treatments for patients with diseases like melanoma and bladder cancer and lung cancer that have just totally transformed these diseases in the last 4 or 5 years. And the basic observation, and one that turns out to be very important, is that cancers live in neighborhoods, right? Now this is a really, like most ideas in medicine, this is actually a really simple idea that we ignored for a very long time. For a very long time, whenever I would look at a cancer as a doctor, patient would come in with a breast cancer, we would look at the characteristics of the cancer cells, but would ignore all of the other folks who were living in the neighborhood, right? This is kind of like if I'm a policeman and I go into a neighborhood and the only person I care about in the neighborhood is the criminal, and I don't care about anyone else in the neighborhood. Okay, which ends up with not very good community policing, as we all know. So the question that came up starting about a decade or so ago was, could we enlist the neighborhood watch to help fight the cancer? In other words, not just kill the cancer cell, not just take a baton and hit the criminal over the head with it, but actually use the strength of the neighbors to help take care of the cancer. Okay? And it turns out that if we look at your body's immune system, the immune system has lots of on and off switches. Now, I was asked for decades by cancer patients, I have breast cancer, is there something wrong with my immune system? And my answer was always the same, no, your immune system's great. If you come down with the flu, you have exactly the same chance of getting sick as I do, okay? And not for the first time in my life, I was absolutely wrong. Okay, because it turns out that in the neighborhoods where cancer cells are, there is something wrong with the immune system in many, many different cancers, okay? And this is another one of these really incomprehensible graphs that scientists like to show. But if you think on the right, in the pretty pale blue, is the cancer cell, and on the left is what's called a T cell. Now, T cells are white blood cells, and they're white blood cells that fight cancer. And they circulate around in the blood, they go into human cancers, they become part of the neighborhood, and in many cancers, they represent sort of the body's surveillance against cancer, and they keep the cancers from growing. They actually attack the cancers, eat the cancers, okay? And that's what T cells are supposed to do. But I told you that T cells have on and off switches, and here's the problem. Cancers are smart, and one of the things that cancers can do is that they can flip the switch, okay? And they do this by a protein that's found on the surface of the cancer, shown here. It's called PD-1. If PD-1 hooks up with PDL1. If PDL1 hooks up with its receptor on the T cell, what's called PD-1, that flips the switch. And it tells the T cell, don't attack the cancer cell. Okay? Now, I don't know if that's confusing enough for you or not. So, okay, show of hands for those of you who are Star Trek fans. Star Wars, pardon me. I'm sorry. I write blasphemy. Okay, Star Wars fans. Okay. So let's see if I can get this to work. Yes. Okay. So here, this is exactly everything you need to know. Whoop, that didn't work. Let's try this again. Okay, you're probably smarter this than me.
Let me see your identification.
You don't need to see his identification.
We don't need to see his identification.
These aren't the droids you're looking for.
These aren't the droids we're looking for.
He can go about his business.
You can go about your business.
Move along.
Move along. Move along.
Okay. That's what a cancer cell does to a T cell. Okay? Yes, and that.
Possibility to test if the cancer cell that whatever binds with the other
Yeah,
one,
absolutely.
how much we have that in our body.
Yeah, so there's, I mentioned these things called PD-1 and PD-L1, and in the laboratory, and indeed in clinical samples where we use these drugs regularly, one of the things that's measured is do you have PD-1 or PD-L1 in the tumor? So a pathologist can measure it pretty easily. Yeah, absolutely, yes.
Is the reduction of protein consumption, does that correlate to what you're talking about in terms of the cancer?
So this is something very specific, and it's not related directly to protein consumption. It's related, so T cells always have this protein on it, the PD-1 protein. It's a normal receptor on the T cells. What's abnormal is when the cancer cell has the PD-L1, the thing that flips the switch on the surface. Some cancer cells have a lot, some have a little, and some have none. Okay, and part of how dangerous a cancer can be is related to how much of this protein you have on the surface of the cancer cell. A lot on the surface of the cancer cell, and your immune system goes to sleep. Okay? Yes?
Is that something that's routinely looked at, the pathology of the cancer tumor, or is that a special test?
So, well, it's a common test in some cancers. So, for instance, in lung cancer, the FDA approval for what's called the checkpoint inhibitor drug that we use, and you can see this on TV. I've even seen an advertisement for this on TV. If your tumor expresses a certain protein, I think you may have seen the advertisement on the TV. Actually, what the protein they're talking about is this protein. Okay, and so yeah, in some cancers it's measured regularly. These drugs are approved for lung cancer, they're approved for melanoma, they're approved for bladder cancer, they're approved for head and neck cancer and a few other cancers. They're not yet approved for breast cancer because we just simply don't have as much data, and we're not quite as comfortable with the efficacy that we are in the way that we are for the other cancers. And I'll get to that in a minute. Okay?
But it seems
Yeah.
that regardless of, to do with the drugs,
Yeah.
that when they do a pathology on a breast cancer tumor, seems to me, it would be good to know if it had this, because then that means that your own T cells are not doing the job, will not be able to do the job that we might count on them to do.
So it's a great question, and if you didn't all hear it in back, the question is basically, shouldn't we be measuring this in every tumor? Okay, and here's kind of the interesting answer, which is that if we look at breast cancers, some breast cancers have a lot of T cells in them, and some breast cancers have no T cells in them. Okay, so not only do they not have the receptor that you can measure on the T cell, a fair number of breast cancers don't even have any T cells in them, or have so few in them that it doesn't make any difference. Okay, now the term doctors use is TIL. TIL stands for tumor infiltrating lymphocyte. I think it's basically these T cells. And TILs are really common, or pretty common, in triple-negative breast cancers, and they're pretty uncommon, for instance, in estrogen receptor-positive cancers. Okay, and indeed, this has led the people who are studying these new drugs to focus more on the triple-negative breast cancers than on the ER-positive cancers. HER2-positive cancers fall somewhere in between. This hasn't been a standard thing to measure by pathologists. Pathologists will start measuring it when we start having the studies that show who it works in. Okay. So this is sort of a glass half empty, glass half full story. So let me show you two different studies that were presented just recently at the American Society of Clinical Oncology meetings, because this will give you some idea of what we're wrestling with for patients with advanced breast cancer. One is a study called the Keynote Study. And the Keynote Study used a drug called pembrolizumab. Doctors tend to call this Pembro. In patients, by and large, who had metastatic triple-negative breast cancer. And very large study, involved well over 100 patients. You got Pembro for up to 2 years if your tumor appeared to be responding. Now here's the problem. If you looked at this more than 100 or so patients, only 4.7% of the patients had a major response, what we call an objective response to pembrolizumab. In those patients, it worked really well. So if you got a response, you could go into remission for years. But 95% of the patients didn't get a response. Okay? And we don't know why, by the way. When we've done laboratory tests to try and figure out who responds and who doesn't, turns out we don't have a very good idea. So that's the glass half empty. Okay? If we now take exactly the same drug, pembrolizumab, and use it to treat someone with newly diagnosed breast cancer. And for many patients with newly diagnosed breast cancer, we now give our systemic therapy before the surgery. It's what we call neoadjuvant or upfront therapy to shrink the tumor and make it easier for the surgeon. So what happens if you use Pembro in someone who has a locally advanced breast cancer, a large primary breast cancer? Well, what happens is if you have a triple-negative breast cancer, you triple the number of patients who have a pathologic complete response, where when we go to surgery, we can't find any cancer in the breast or in the lymph nodes. Okay? So on the one hand, I told you about advanced breast cancer, where we got a 4.7% response rate, which is disappointing by any standards. On the other hand, I told you about a tripling of triple-negative breast cancer. And by the way, the absolute numbers here are, it went from a 20% pathologic complete response rate to a 60% pathologic complete response rate. Okay? So that's a really exciting number for doctors and hopefully for patients as well. And yet the same drug for patients with advanced disease was kind of a stinker. So we're still learning a lot here. And this latter study, the glass half full study, is one where we don't have long-term follow-up. So we don't know whether or not this tripling of pathologic complete response rate is going to mean that we're going to cure more people. We're hopeful that it will, but we don't know that yet. So we're kind of at a point yet where we're still learning a lot about these immune checkpoint inhibitors. And hopefully we'll learn enough to be able to identify who benefits and carry it forward. Why might there be some of these differences? Well, interestingly, primary tumors tend to have more of these TILs, the tumor-infiltrating lymphocytes, than metastatic tumors for reasons we don't quite understand. In the primary setting, we were giving the antibody along with chemotherapy. Maybe there's something special about the combination. If so, that might be good for patients with advanced disease as well. So does that combination equal benefit? And then finally, what does this mean for trials for patients with early stage disease? We don't know yet. But there's a bunch of trials that have been initiated, including some very large randomized trials in both the advanced disease and the early disease setting, to see whether or not we can actually use these drugs to improve the fate of patients with triple-negative breast cancer. There's also some trials, not to throw things away, some trials that are being looked at in ER-positive and HER2-positive breast cancers as well. So we hope we're going to have some more data on that in the not-too-distant future. So with that, I'll come to an end. There's a lot of things I thought about talking about tonight, and perhaps in the Q&A we can go into some of those things. But hopefully what I've done for you is to give you some idea, first, that breast cancer is truly a family of diseases. I'm frequently asked, Dr. Sledge, when do we get to cure breast cancer? And the answer is, we're not going to cure breast cancer. We're going to cure breast cancers, okay? Because there isn't one of them. There's a bunch of them, and we need to find better treatments for all of these different cancers, okay? We're getting there, and we're getting there in large part because we're learning more and more about the biology of the disease. I've been in this field for a very long time, as you can see by the fact that my hair is no longer its beautiful dark brown that it was when I was an oncology fellow. I'm pretty confident that over the next decade or so, we will unravel most of the remaining secrets about breast cancer. We have learned so much in recent years as a result of our understanding of the genomics of the cancer, how the DNA affects the cancer. I can barely open a scientific journal without seeing an article about something new and something exciting about the biology of cancer. Clearly, we're going to see real advances over the next few years, and hopefully they'll be in a timely fashion for several of my friends here in the room. So with that, why don't I open things up for a Q&A? Yes, ma'am.
With breast cancer, one goes into genetic testing.
Yeah.
And they say we test for the known genes and the mutations. But what, 32 or 36 of the known.
Yeah.
And then recently something was posted that there are more studies that have come by. By when would those be available for retesting?
So the question is, if we look at genetic testing, I guess in a way the question is, how do we know what to test, right? And what to test for. Now, if you were to go back just 3 or 4 years ago, we tested exactly 2 genes. We tested BRCA1 and we tested BRCA2. And the reason for that was that starting about 1995, when 1994, 1995, when BRCA1 and BRCA2 were identified by scientists, a company called Myriad Genetics in Provo, Utah, got the patent rights to your chromosome 14 and your chromosome 17. Okay, no one else could test for them. Okay, crazy, right? It's your gene, not the company's gene. But the patent office had actually said that this particular company owned chromosome 14 and chromosome 17 in your genome, and no one else could test. No one else could test it. Okay.
Or how did they take ownership of those?
Pardon?
Did they discover it, or how were they?
The funny thing is, no, they didn't discover it, but they were the first people to apply to apply for the patent. Now, the Supreme Court, which for once had a thoroughly undivided view of this, voted 9-0 3 years ago to overthrow the patent. So you now own chromosome 14 and 17 again. Congratulations, right? Okay. And that plus all of the genomic stuff we learned in the meantime led to kind of a race to the bottom in terms of the cost of doing this. So whereas on average this cost $3,000 or $4,000 3 or 4 years ago, it's now down in the hundreds of dollars, and it's getting cheaper every year. And as it's gotten cheaper, and as we've learned more about the genetics of breast cancer, we actually have started to test for more and more things, and more inherited mutations. So we now do what's called panel testing. And in panel testing, we look at not just BRCA1 and BRCA2, which are the 2 big dogs, the most common ones, but also start looking at rarer things that we know also are associated with increased risk of breast cancer. And in the broader panels, we're now actually looking at cancer genes that don't just predict for breast cancer, but also ones that predict for colorectal cancer, uterine cancer, you name it. Okay? And when you do that, you find some interesting things. First off, we find that the sort of spectrum of mutations that are associated with breast cancer is much broader than what we used to. But the real important part of this, I think, is pretty much what I told you in the talk, which is that we're actually going to, in the future, going to be giving treatments based upon what sort of cancer mutation caused your cancer. I mentioned the PARP inhibitors for BRCA1 and BRCA2 tumors. It's not at all impossible that we'll be using these tests to say you should be getting drug X, you should be getting drug Y going forward. And I think the other part of this is that as this gets really, really cheap, well, hopefully that will also mean that we'll be able to find out who's at high risk at an earlier point in their life and intervene and actually prevent them from developing cancer, which I think, listen, my goal in life has always been to be out of a job before I retire. okay? You know, anyone who's a medical oncologist and spends his or her life pumping poisons into people's veins gets tired of it in a hurry. Okay, we love our patients, but we frequently have to give genuinely horrible things to people to allow them to live longer. I would like nothing so much as to be out of a job before I retire. And the best way for us to do that is to find out who's going to get cancer and then find out how to prevent it. Okay, and so that's you know what we as a field certainly need to work on. Okay, yes, ma'am.
How does one get that panel? Is that related to being in studies at Stanford?
Nope. The panel you can you know any doctor can order the panel test. Yeah, you don't need to be at Stanford. And as I mentioned, these tests have gotten cheaper. So there's a local company. It's called Invitae. It's in San Francisco. It's one of the 5 or 6 companies that does this genetic testing now. Their goal is to do the entire what's called mendeliome. The mendeliome is every mutation that causes an inherited disorder, okay? Their goal is in the next 2 or 3 years, they want to do the entire human mendeliome for, I think it's like $400, something like that. That's what they would like to do. By the way, when I say the entire mendeliome, I don't mean just cancer genes. I mean genes that cause heart disease and kidney disease and lung disease as well, okay? The price of this stuff is getting really, really cheap. Yes, ma'am.
Is it your position then that cancer is largely genetically driven?
No, it is not. So the question is cancer largely genetically driven? Actually, let me qualify that. Yes, cancer is almost 100% genetically driven, but it's not 100% inherited mutations. Okay? So in breast cancer, you know, almost all breast cancers have something funny about their DNA. Okay, they almost all have some mutation in the DNA. But in most cases, you actually didn't inherit that. In most cases, it was something that happened during your lifetime that caused the cancer. Okay, so here and then here, okay?
Are the drugs that are commonly used to treat estrogen-positive tumors as successful with predominantly progesterone-sensitive tumors?
Okay, so the question's about the progesterone receptor, because you know when we test breast cancers, we routinely test not just for estrogen receptor, but also for progesterone receptor. This turns out to be a total historical accident. I mentioned earlier that there were 700 different proteins in your body that are regulated by estrogen. What was the first protein that was detected that was an estrogen-regulated protein back in 1976? Progesterone receptor, okay? So we could have picked any of the other 699, and it probably would have worked equally as well as progesterone receptor. But when you measure progesterone receptor in a breast cancer, what you're measuring is the ability of estrogen to affect the cell. Because if the progesterone receptor is there, it's probably there because estrogen was stimulating it to be there, okay? So if a patient has a tumor that is estrogen receptor positive, but progesterone receptor negative, that tells you that there's something a little bit funky about how the estrogen receptor is affecting the cell. It's not doing what it's supposed to do. And actually, one of the things it's supposed to do is to stimulate progesterone receptor. Mm-hmm. Yes, sir?
My question is, in your example, you basically using each drug to block each of the pathway.
Yeah.
The example of
Yeah.
20
Yeah, yeah.
But if the disease progresses, you move on to stop giving that particular drug, like Herceptin or Tedium-1, and you give a new medication, right?
Mm-hmm.
So if you stop giving by giving Herceptin, isn't the cancer is going to still go back the other route?
So it's a great question. So the question is, let's say that you have let's take our 2 receptors, estrogen receptor and progesterone receptor. We have tumors that are both ER positive and HER2 positive, okay? So both HER2 and estrogen can stimulate the growth of these cancers. So let's say, shouldn't I be blocking both? I think is the question. And the answer in the clinic is that if I have someone who has early stage breast cancer and I've finished chemotherapy and I'm giving them Herceptin, I'm also giving them a drug that blocks estrogen. So yes, the answer is yes, I do both of those, okay? But I think the deeper question that you're asking is, as we have all these new targeted agents come along, and you know, as I said, we throw up a roadblock, and the cancer finds a way around the roadblock. Well, shouldn't we still have that roadblock up when we go to
Yes.
the next drug? Yeah, and I think it's a good idea. Now, the problems with it are sometimes biological and sometimes practical. Sometimes these drugs don't mix and match very well. So some of these drugs, when you combine them, you end up with a lot more side effects. So that's one problem that we see sometimes. The other problem is the obscene expense of modern cancer drugs. Most new drugs coming on the market today cost about $10,000 a month. Okay, I mean this is this is ethically dubious. It's insupportable economically, and it's going to eventually drive the healthcare system into financial ruin, and it already drives patients into financial ruin on a regular basis. Healthcare is the leading cause of bankruptcy in the United States today. Okay, I'm sorry, that's a rant and away from your question, but financial toxicity is as real as medical toxicity for many patients. Okay, so yes, that's definitely an issue. So in back and then in front, yeah.
You mentioned two drugs working well together. You mentioned earlier the Monarch trial. You said
Yeah.
it was just recently FDA approved.
Yeah.
Does that trial work well with the aromatase inhibitors?
So yes, so the answer is yes. The trial that I ran was with another drug called fulvestrant or Faslodex, but the same drug has also been combined with aromatase inhibitors, and both with aromatase inhibitors and with Faslodex, combining the hormonal therapy plus the spanner in the works, the CDK6 inhibitor drug, provides benefit. So yeah, we routinely use those in combination.
Now that FDA approved, how soon are you going to be prescribing it to your patients?
Oh, I'm already prescribing it to pay. Yeah, we routinely use these drugs now for patients with advanced breast cancer. What we're not using them in yet outside of clinical trials is for early stage breast cancer. And that's in large part because we just don't know if they work there. So those trials are going on. My hope is that in a couple of years we'll have an answer to that question. If the answer is yes, then we'll be using them a lot in patients with early stage breast cancer. Yes, sir.
You mentioned a chemical that was put on the shelf because it was too toxic.
Yeah.
I was wondering, why did they put interleukins on the shelf?
Well, interleukins are pretty toxic. Actually, we still use interleukins for some cancers. So, for instance, interleukins have been used for melanoma and kidney cancer, and they're FDA approved for both. What's happened though in recent years is that the checkpoint inhibitors, the new drugs that I was talking about when I was talking about immunotherapy, those are hugely more effective than interleukin, and while they have their own side effects, they're hugely less toxic than interleukins by and large. What's called high-dose IL-2, which is the major interleukin that we've used to treat patients, routinely fetches you up in an intensive care unit when you give it. In fact, we administer it in an intensive care unit, so it's pretty toxic. So the newer immuno-oncology drugs are a whole lot safer than the interleukins, and it turns out they're a whole lot more effective as well. So I remember there was a hand in back. Did I see? Yes, ma'am.
Yeah, we keep often hearing that vitamin D is linked with, low vitamin D can be linked with cancer. If you're having a lot of inflammation, it's not good for the body. To hear it from you, I mean.
So the question is, I guess the larger question is vitamins in cancer. Vitamins had kind of an interesting role or an interesting look at in cancer studies over the years. There's a lot of data from the laboratory that says that if you add certain vitamins to certain treatments, and an example is vitamin D added to chemotherapy, that increases the chemotherapy's ability to kill the cancer. But it's been a whole lot harder for us to show that in the clinic than it is in a test tube for reasons no one quite understands. Also, if you ask the epidemiologist, and one of the classic examples of this is vitamin C and vitamin E, okay? There's a lot of epidemiologic data that if you had good levels of vitamin C and vitamin E, you had a lower level of developing cancer. If you had very low levels, you had a much higher risk of developing it. This actually led to the National Cancer Institute to fund a very large trial that was done in Finland, of all places, okay? Now, it was done in Finland for an interesting reason. At the time, Finns smoked like a furnace, and they ate a whole lot of smoked fish. So they had high rates of lung cancer and stomach cancer. So the NCI said, oh, this is a great place to test to see whether or not vitamins make a difference. So they took a bunch of Finnish smoking men. Okay, you have this vision now of a room full of smoking Finns, okay? and gave half of them high doses of vitamin C and vitamin E, and the other half they gave them a sugar pill, a placebo. And they followed them for several years. And it turned out that there was a statistically significant difference in the rate of cancer between the folks who took the vitamins and the folks who didn't take the vitamins. Here's the problem. It was in the wrong direction, okay? All right? They actually ended up with more lung cancer, not less, okay?
More vitamins?
Yeah, the more vitamins actually equaled more cancer there. And, you know, this taught doctors to be really careful, you know. And so I'm frequently asked in the clinic, you know, should I be taking this, should I be taking that? And my answer is always the same, which is I can only tell you what I know. And I can only tell you what is tested. And I tend to be very cautious about making recommendations about things that I'm ignorant about. Because what science has taught us is that our biases are frequently wrong. And even our evidence that we had in the past, when we put it to the fiery test of a phase 3 trial, a lot of our preliminary data turns out to be wrong as well. So we like to be very careful about it. It always impresses me. I say this both as a doctor but also as a patient, that people are frequently much tougher on their auto mechanics than they are on their doctor when they're asking for evidence about something. Okay. And the answer is you should be pretty tough on the evidence, actually, whether or not it's coming from a doctor or from a friend who tells you should be taking this, okay? You should always ask, what is the evidence that this actually works, okay, as opposed to what's your bias or what do you believe. Belief is not very useful in terms of choosing medicines. Studies are pretty useful. Yeah.
I think we have time for one more question. Anybody have another question?
Oh, I can ask.
Sure.
Immunotherapy, how far you think, in your opinion, we far away to get immunotherapy for breast cancer? Like, is it 1, 2 years, 3 years?
Don't know, because I don't know. So the question is how far away for immunotherapy and cancer, and the answer is I don't know because I don't know what the results are. The results will be. There's a bunch of phase 3 trials going on. So they're like, at latest count, I think 5 different companies that have FDA-approved immunotherapy drugs.
For breast cancer?
For all cancers, okay. And pretty much all of them are testing it in breast cancer right now, and several of them are testing it in phase 3 trials. Phase 3 trials are the big, large, definitive proof of concept trials that the FDA requires before a drug goes on the market. And so some of those trials will have the answers in probably the next couple of years. So hopefully we'll have some answers. Hopefully they'll be good answers. But right now, it's anyone's guess as to what the answer will be. I should mention, though, that you know, I showed you that slide with the T cell and the cancer cell, and I was talking about the on and off switches, and I pointed to one box. Did you notice that there were like 10 or 15 others up and down the T cell? Okay. And by the way, that's a very simplistic one. More complicated ones have another set on the other side, because there's both on switches and off switches for these T cells. Okay. And we're just starting to look at these things. And, you know, we've looked at the first 1 or 2, but trust me, there's like 10 or 12 others that are being actively investigated. And we now have drugs coming into the clinic that are looking at both the on and the off switches for the T cells. So we're early days yet. You know, this is like talking about the future of automobiles in 1902. Okay, we know that there are automobiles, but you can't imagine driving from New York to California because there's no interstate highway system, right? But pretty soon, there will be.
Thank you.
Mm-hmm. Great. Thank you all. Mm-hmm. Mm-hmm.
Wow, well, thank you very much, Dr. Sledge. And I'd like to thank my colleague also, Esther Chien. I don't know if you have all met her, but we put these programs on about 6 times a year. You know, half of them are done here in the health library and the other half are done in South Bay. So keep your eyes open. We do have other programs that are coming up. If you haven't picked up our calendar of events, please pick one up. These are really great wellness programs, and you've already heard Dr. Sledge, and if you've come to any of our other lectures, the evidence behind self-care, you know, meditation, yoga, these have all been shown to really help not only with pain control, but stress reduction, and have even shown benefits in extending your life. So take advantage of them. They're free to you. They're free to your caregivers. And we really, you know, we have a lot of amazing programs that are around in the community, not only here in the Palo Alto area, but also in the Los Gatos area. And we also have a yoga or a Pilates program in San Carlos. So take a look at those. I'd also like to let you know that we have our next lecture will be in January. And we have a spiritual care counselor who is also a financial planner. So I don't have the exact title yet, but it will be on estate planning and financial planning. And so it'll be a panel of financial planners that can kind of help you sort through some of the insurance logistics and also some estate planning if you'd like to. But, you know, it's really kind of taking the mystery out of all the insurance things that you're going through with your cancer therapies and your life. It'll be in this library. Again, it'll be on January 24th, but you'll be able to find it. Our new calendar will come out January 1st, and they're good for 6 months. But, you know, our exercise programs pretty much stay the same. We have some that you have to register for, but then we also, you know, have some that are just drop-in only. So go to our website. That's in the front of our calendar as well, if you haven't used it. At the bottom of our website, you'll find all of our lecture series that have been videotaped over the years, and I think we have, 35, 36 lectures that are there for you to review. And this one is being videotaped this evening and will be available in a few weeks on our website as well. Okay? We also, one more thing, we'd like to know what you thought of tonight's program. So we have an evaluation form, if you don't mind taking just a couple of minutes to fill that out, and also let us know what you'd like to see in the future, because your opinion is really valuable to us. And I, again, want to thank you for taking the evening and spending it with us. Okay? Thank you.
Thank you.